Let me, okay, got it. Let me start the share screen. Share screen, there it is. You should be seeing my screen. First of all, I'm going to press play, and then I want to remove the floating controls. There we go. And I'm then going to move this. Great, wonderful. So, great. Thank you very much for showing up to the second lecture in my series. And as usual, while people are drifting in, I'll give a brief review of what we said in the first lecture. So remember, in the first lecture, I was introduced the idea that computation in the brain is distributed. Computation is done by neurons, of course, individual neurons, but it's not like you have this gigantic, either a von Neumann architecture or an undifferentiated sort of neural network. Rather, at the biggest scale, all the different functions you want to perform are distributed across various kinds of brain areas, which do specific things. They contain algorithms that do one thing versus the other: motor control, speech, vision, smell, etc. So the computational function is distributed across. If you pick any one of these areas, then it's distributed across that area with specific circuits doing specific things, in this case, controlling the body. And if you really, focus in one more step into each one of these little areas, it's the diversity is kind of insane. As in, the retina alone contains 60 different types of cells, illustrated here. And one of my key questions that I tried to raise to the group was, why would you design things in such a manner? Of course, there could be historical reasons, right? Evolution is history, or there could be a design reason, as in, it's selected for some purpose. And I tried to argue that if you ask the question, why does the brain distribute computation and what organizational principles organize the circuits at each scale, that it's reasonable to think that one principle that selects this kind of diversity is efficient use of resources. That is to say that brains may exploit the average structure of the world to efficiently allocate limited resources to maximize gain for the organism. Many things to be defined there. What is resources? What is gain? What is computation? What is communication? But the reason for this hypothesis is there's reason to believe that the brain is expensive tissue and that you need to invest it well. I also mentioned that there's definitely not the only principle that's operative. There are other principles too. For example, one thing that certainly happens is that you have the fixed structure that you get through development, but then the circuits that you get self-organize their architecture and dynamics through learning in order to adapt to variations in the world, to learn new tasks, correct inaccuracies, etc. We didn't spend any time discussing this in the previous lecture, but I will in the latter half of today's lecture. Again, this is a very big field, and these are all, summer school lectures are supposed to be an opening into areas. So you should find what you're interested in and then go read more, because there's no way any given lecturer, myself included, is going to cover everything that is of relevance for any one of these topics. Great. So I tried to justify to you that this is a useful way of thinking by talking about two kinds of things. One thing that I did was to discuss, the overall flow of information out of the retina and how it's partitioned between the different cell types. And then to try to drill down and give more specific examples, I pointed out that this kind of idea, that efficient partition of resources, has been applied very powerfully in the visual system. And this is just a sampling of the papers, and these are just the papers from my own group, but lots of people have written about this kind of thing. And I focused on two examples: the proportions of red, blue, and green cones, and on the bright and dark proportion of bright and dark detectors in the retina, and tried to discuss those things which are otherwise mysterious in this language. I then mentioned that texture perception, a cortical texture, which is mediated by cortical processes in V2, in the second layer of visual cortex, is also an interesting sort of cortical example of this kind of idea of efficient partition of resources. In other words, that these ideas are not just useful in the sensory periphery, but also deeper in the brain. So today I propose to do two things. First, I propose to drill down further in these sensory examples by doing something I didn't do for the other group, which is for the other group, I explained these ideas in the visual repertoire some more. But I'm proposing for you to instead talk about examples in olfaction, spend a little time doing that, and then go on to the main topic of today's lecture, which is to discuss a deeply cognitive thing, which is your mental representation of space. Let me first pause to ask, were there any questions from the previous part, which was a recap of the last lecture, that you would like me to address right away? or can I move on to talk about olfaction? Okay, I will move on. So, it occurred to me that it's probably so much of our imagination about problems in theoretical neuroscience is really governed by our experience with vision. We're very visual animals, and vision is the most studied sense. So a lot of our thinking has really been driven by thinking about vision. So it's really useful to think about systems that are very unlike vision in order to see if the principles you're trying to develop have any general applicability, or are they secretly just principles that apply to the visual system. So to this end, let's think a little bit about the olfactory system. Here's a famous drawing of the early olfactory system, also by Cajal. So I've written down here a list of papers that my group has published in the recent past, based on which I'm going to give this lecture. So I want to start out by thinking about, remember when we discussed how the visual system could be adapted to expend resources well. Really a key step there was to ask, what is the visual system trying to process? We had to know something about the statistics and structure of the natural visual world, and then we could reason about how the circuits that process it should be structured. So we should start out by talking about the structure of the natural olfactory world. Now, the olfactory world is a lot more complicated in some ways than the visual world. There are many, for starters, what is olfaction about? Well, it's about sensing volatile molecules. And there are at least 10,000 different kinds of volatile molecules, right? So basically stuff that can float in the air and that could be sensed. Some people will tell you there are 100,000 or nearly a million different kinds of volatile molecules, but there are at least 10,000 different kinds of volatile molecules. To make matters worse, if you want to sense these things, typical natural odors are mixtures of at least about 50 molecular species. So, as a rough count, if you have about 50 molecular species, there are 10,000 to the 50 kind of natural smells that you could imagine making. That is an insane number. It is so enormous. Even imagining how you can make a system to process these things is hard. Now, and what's more, the odor environment changes, reflecting changing seasons, circumstances, new opportunities, and new threats. And that circumstance means that you might have thought that the way you deal with this 10,000 to the 50 possible natural smells is you ignore most of them. Well, I don't care, right? All these smells, but I just care about banana and coffee. You could imagine just caring about a couple of things. But that's a very bad idea, because if you're out there, then if I were your predator, and you had a hole in your olfactory sensory space, if I were your predator, I would go hide in that hole. And then you wouldn't be able to smell me, and I would eat you. So it's a very bad idea to have olfactory holes, I would say. And it's important that you be able to kind of sense, one way or the other, the entire space. So what's interesting is really olfaction is in some sense a sense of touch. It's a method for feeling the shapes of molecules, because smell is really about the shapes of molecules. So you have a combination of 50 odorants. You want to feel their shapes and somehow come up with the percept. So how do you do this? So of course, you have a nose to do this, and odors are sensed in the nose by olfactory receptors. So when molecules bind to an olfactory receptor, the receptor responds, and you have the sense of smell. Now observe that it turns out that Linda Buck and Richard Axel won the Nobel Prize for determining that every olfactory receptor type has a separate gene, right? So it's all encoded by separate genes. There are about 100 in fly, well, maybe 60, and there are about 300 in human, and 1,000 in mouse, and dog and elephant, with this gigantic nose of its that it has only about 2,000. So you might say, oh, that's pretty different, right? It's an order of magnitude different. But I would claim to you that these are almost all exactly the same. Taking human in the middle, fly has about 1/6 as many receptors as human, and elephant has about 6 times as many receptors as human. Order 1 factors compared to human. Everybody seems to have a few hundred receptors, which is pretty nuts given the range of body sizes we're talking about. The elephant is 1 billion times heavier than the fly. right? And usually you have scaling laws that tell you things increase with size. So that's weird. There's an operating point of a few hundred receptors that basically all animals have. And I've already argued to you that you can't have holes in olfactory space. So in this context then, there's a very strange thing going on here, right? You have an odor space that's high dimensional. So if I think about the odor vector as a vector of concentrations of different odorants, it's like what, a 10,000 dimensional space at least. And you have a low dimensional response space, about 500 dimensions. And I'm supposed to faithfully represent the high dimensional odor space and the low dimensional response space. How do I do this? Now, this is to draw a physics analogy. This is like telling you you have three-dimensional space, x, y, z, Cartesian coordinates, and I insist to you that you must encode these three dimensions in one coordinate x. Well, that sounds crazy. Of course, you could do it, right? You could run a space-filling curve through these three dimensions. And if you did, you'd be able to read out the coordinate in the space-filling curve, and that would give you in one dimension. But that would be a crazy representation because nearby points in three dimensions will get mapped to very distant points in one dimension. And so you're not going to be able to tell apart very easily what's similar and what's different. And of course, clearly, one key function of a sensory system is to say what's similar and what's different. So what you would like is an encoding from high dimensions to low dimensions that preserves the distance between odors. Okay. So, so there are, in the context of our, efficiency and distributed computation, there are a bunch of questions we want to understand, right? Why do you distribute the olfactory information in a few hundred receptors as opposed to more or less? How do you distribute the information in a manner that preserves the required invariance? Is there any evidence that this distribution is somehow done efficiently or effectively? And then how can you decode it? See, these are all questions that you might be interested in asking in the context of our broad hypothesis about distributed computation. So let's start digging into this. So what's the geometry of the space of olfactory mixtures? So if these are all the molecules going this way, and the odors, these are all the different odors, but odors, I mean odor objects, combinations of molecules. Then, odor one may contain, odorant, the first, the third, and, whatever, this is the seventh odorant, they're all mixtures of different kinds. But the thing I'd like to point out is that no animal or plant makes an odor composed of 1,000 molecules. It's expensive to sit down and make molecules, right? It takes effort, and you have to commit resources to it and just throwing the stuff out in the air. So it turns out that if you look at foods and fragrances and stuff like that, flavors, etc., there are about 40 molecules, 40 to 50 molecules. And so in the very real sense, every odor, if I think about it as a concentration vector, is a sparse vector in chemotopic space. So out of the n-dimensional space of possible molecules, a natural odor will be sparse in the sense of containing k much less than n components. Call it about 40. So you might draw that the following way. So if k was 1 and n was 3, you would say, well, the first odor lives along the z-axis, the second one this way and this way. In other words, basically the structure of odor space is like a union of hyperplanes. Right? Each hyperplane, which is a combination of odorants, defines an odor object, right? And different species of that flower or different individuals of the flower may have slightly different combinations of those different odorants, but they still have those odorants in them. And then you have this union of hyperplanes. So this is the thing that you want to somehow represent. That's the leverage you have. This is the analog. This structure is the analog for olfaction of the fact that in vision, you have these sort of correlations, long-range correlations in the pixel intensities, and you have regularities in the intensity distribution, etc. This is the structure you have. So this is what the system can leverage. So suppose you have data like this. Now I'm going to think like a theorist, right? I'm not going to ask what the olfactory system does. I'm going to say, I give this problem to a computer scientist or a mathematician and say, well, you have this sparse chemotopic space. How would you encode it? And a standard answer now would be, well, here's the thing you can do. You take this long sparse vector, you multiply it by a matrix, which can be a random matrix, by the way. It can be a completely Gaussian random matrix, or it can be a structured matrix with a certain property known as the restricted isometry property. Okay. So if you multiply it by, let's say, a random matrix, you'll get a much smaller, denser vector. And there's a theorem that says that so long as this big vector, let's say the odor vector, is sufficiently sparse, and this sensing matrix, if you like, is sufficiently unstructured, then using many fewer measurements, M here, so with these are receptor responses, if you like. So each of these rows tells you the response of one receptor. This is one receptor. With M receptors, where M is much less than N, actually M is about K times the logarithm of N is what it has to be. You can completely, the small vector contains all the information necessary. It loses no information from this big vector. And what's more, the theorem also says that distances between odors are preserved. So in other words, if you compute the Euclidean distance in this chemotopic space, it's going to turn out to be the same as the Euclidean distance between this small space. So that's exactly what we wanted, right? We want to take this gigantic signal, compress it into a much smaller receptor response space, and we want to preserve distances. So it seems like, wow, something like this would be a good idea for something like this to happen. So just to say it again, the typical odor contains maybe 50 of the tens of thousands of molecules. If each receptor binds with quote unquote random affinities and basically unstructured affinities to volatile molecules, the idea would be perhaps you need only a few hundred sensors to represent all the odors, right? And indeed, if you take 50 times the logarithm of 10,000, you're going to get numbers of the order of a few hundred. That's the idea, right? So the question is, does the olfactory system use this approach? So in order to study this question, so this is going to try to come up with a theory of why you only need a few hundred receptors. Notice that's where I'm trying to go after, about why you distribute information in a repertoire of this size is what I'm going after. And I've tried to argue to you that already qualitatively, the ideas of compressive sensing suggest that you can do this with a few hundred sensors. So let's try to get some evidence that maybe something like this is going on. First, what we need is a model of the response of receptors, right? We need to be able to handle that. And we need a model basically because, suppose I take mouse, there are 1,000 receptors, there are 10,000 odorants. What you really want is somebody to tell you for every odorant, how does every receptor respond? So, that's a matrix, that's, if you like, 1,000 by 10,000. No one's made these measurements, right? The kinds of panels you have is you have 100 odorants and people will tell you the answers of about 50 receptors. So you need a way of sort of understanding this more broadly. So here's one way of thinking about this. You can do classic biophysics. You imagine you have a receptor, and the receptor, let's say, can bind in this case to the green molecule or the red molecule. There are various kinds of binding affinities and rates of going back and forth. And you can work out in such a circumstance, what is the response function of the receptor going to be to mixtures of molecules. So the really hard part is to work out what are these mixtures of molecules, because even if you could measure, for example, the response of receptors to a panel of 100 odorants, trying to work out the responses of the receptors experimentally to mixtures of the 100 odorants is a combinatorially impossible task, right? So if you have only, for example, a mixture of 5 odorants, that's already 100 to the 5. If you have 100 odorants, it's just not possible. So you need a biophysically motivated model. So what I'm writing down here is classic biophysics of receptor binding to ligands, and you can work out a simple sort of a competitive binding model and of receptor responses. So this is a very simple model of how receptors are responding, and what this picture is supposed to show based on results in this paper is that this extremely simple competitive binding model, it just says that ligands compete according to their affinities to bind to receptors, gives an excellent description of receptor responses. The reason I'm presenting this is for photoreceptors, which is what we discussed earlier. This has been done, right? People understand in exquisite detail how photoreceptors respond to photons of different frequencies. So we know that. So we need a piece of that. Also for olfaction, and I'm trying to tell you that a simple competitive binding model will do well in serving as your baseline model, and that's what we're going to use. You can simplify things a little bit more if you like, which is that, these kinds of competitive binding models are nonlinear. They have a threshold for response, then they rise, and then they saturate. Well, in the region between threshold and saturation, if you like, you can linearize and then treat this as a linear sensing model with this slope. And then, you just take the linearized, then you have a matrix, you multiply the matrix times the concentration vector, and it'll give you a rough approximation to the response of receptors. That's super convenient, of course, right? Because it's much easier to work with a linear model of that kind than a nonlinear sensing model. And so we'll use this linear model also sometimes. Okay, so what's our schematic of the sensing? This is the analog of saying you have the different photoreceptors processing different colors of light, right? So what happens here is that you've got the different receptors binding to and responding to different molecules. So here's data from Hallam and Carlson in Drosophila. So the picture is you have molecules floating around. The molecules sort of bind to the olfactory epithelium in the nose. In the epithelium, there are olfactory sensory neurons. Each color is supposed to represent a neuron that expresses a different receptor. So there are a certain number of receptors, and they're expressed in multiple sensory neurons. The sensory neurons respond, and then at the second layer, there are these things called glomeruli, which collect the responses of all neurons expressing the same receptor type. So that gives you a representation that aggregates the receptors of a given type. And then there's some processing, some lateral inhibition, and so on. And those things are projected apparently randomly to the olfactory cortex. So that's the structure of it. So I'm using language from mammals, from vertebrates, but I'm going to use data from flies. The reason why that's okay for our purposes is that the structure of the olfactory circuit pathway between is essentially identical between flies and vertebrates. So I'll feel free to mix and match data. Occasionally I'll use mouse data, occasionally I'll use Drosophila data. Anyway, this thing is what would be called the sensing matrix, namely Hallam and Carlson went to 24 receptors in Drosophila, this should have been on this side. This ORN should have been this side. Anyway, so they went to 24 different neurons expressing 24 different receptors in Drosophila. And on this side, they had 100 different odorants. And what they did is the color map here indicates the strength of the response in spikes per second associated with each of these odorants. So if you read this way, it tells you if I go to receptor type 1, then what its strength of response is to each of the 100 odorants that you have here. So the way you describe this is you have an environment concentration vector, you multiply it by the sensing matrix, and that gives you the response vector. There is a question.
Yes, I have a small question. If you say odor index here, do you think of individual molecules, or about this mixtures of order 50 right. molecules which compose an odor?
Yeah, so I will attempt to be consistent that odor means mixtures and odorant means odor molecule. But here, I've of course been inconsistent. And odor index here means odorant index. I'm afraid I will not succeed in being consistent because of the problems with the language. So I will try my best. If it's confusing, you should tell me, and I will fix that. But thanks for that question.
I have a question too. So yeah. I'm wondering, why do we arrange this glomeruli, like this down below the bubble, in this way? Is this just autonomy schema, or is there a reason to arrange them like this?
Why do we, or why does the brain?
Yeah, what does the brain? Yeah.
Well, so the standard story that people tell about that is that you've got many receptors responding to odorants. Let's say you have many red receptors in different neurons. The responses are all very noisy. So because they're all very noisy, the standard story that people tell, and that I will use in a few minutes to talk about how many of these different neurons you should have, that the idea is that the glomeruli aggregated to beat down the noise. So I'm going to, you remember how yesterday in the previous, or 2 days ago in the previous lecture, I discussed the relative proportions of red, blue, and green photoreceptors and how that should be organized? Right?
Yeah, so long wavelength and short wavelength.
Short wavelength, long wavelength, short wavelength. So you can think about these different types of neurons here as being like the L, M, and S cones, red, blue, and green cones. Except now in let's say the mouse, you have 1,000 cones for processing odors. And unlike photoreceptors, which respond to some narrow band, these things bind to lots and lots of different molecules. So I'm going to try to answer the question of what should be the different proportions of these kinds. I'll do the same problem again.
Great, thank you.
Okay, all right. So this is a schematic of our sensing model. So this is a setup for that, the initial part. So I'm trying to first argue to you that using this kind of model, you can in fact compress the odor information into the small number of receptor neurons that you have, receptor neuron types that you have. And then I'll try to argue, given that small number of receptor types, what proportion should you have of each one? So I'm actually trying to build up to the problem that we were doing yesterday. Okay, so first I'm going to try to argue to you that you can, in fact, maybe I should have called this compressive encoding, not decoding. So, so what you can do is you can now imagine here is the measured response matrix. You remember this side are the receptors, on the top here are the odors. So you make a long odor vector, you multiply this matrix with the odor vector, tells you the response. And then you can hypothetically imagine other kinds of, olfactory systems, ones that are scrambled versions of the measured thing, a Gaussian random one, which would be kind of ideal according to the theory of compressive sensing, a labeled line one, where you threshold this kind of measured response. You threshold it so that you just say that a given receptor responds to the 5 odorants to which it's most responsive, that kind of thing. So you sort of different approximations of these kinds of pictures, and you can ask how they perform in storing the information and allowing you to decode it on your computer. So it turns out this kind of labeled line where you erase a lot of where you say it responds to the 5 most important odorants does terribly. The decoding error is terrible. The moment you increase the complexity of your mixtures, it's very hard to decode. The actual measured and scrambled version of the measured receptor response matrix do very well, and they are quite close to the ideal, so if you have a Gaussian random matrix here, it's the ideal random sensor, and it'll give you as much information as can possibly be stored in this way from the odor vector. And as you can see, the measured responses, although they don't look like the Gaussian random, actually function quite close to the Gaussian random model in terms of how well they store the information and allow decoding. Okay, so that's a kind of justification I'm trying to give, that the structure of the responses in this response matrix is basically kind of as good as possible for encoding the olfactory information. That's the argument I'm trying to make. So one problem with doing this kind of analysis is, we're using this little data set with 24 receptors and 100 odorants. And I told you, mouse has like 1,000 receptors and there are 10,000 odorants. So the question is how you take this kind of limited experimental data and then somehow build a model where you can at least study these issues by making a pretend sensing matrix that's 1,000 by 10,000. That's what you'd really like. 1,000 receptors by 10,000 odorants. So you need to somehow construct as a theorist to understand these issues. As a theorist, you need to construct a much larger sensing matrix that extrapolates the statistics of the small one. So you can do that. So you take this little sensing matrix that's there experimentally, and all the sensing matrices in different people's data are pretty small like this. And then you can try to extend it by making a bigger matrix that has the same histogram of firing rates, so the same histogram of numbers, and the same, if you compute the covariance of this matrix, you can impose that the covariance looks the same, so that the statistical correlations between pairs of responses look the same. And I'm just telling you that we did that. There are techniques for extending matrices like that, so we use some of those techniques for extending the matrix, and this way, wrote down a synthetic artificial, sensory system, if you like, olfactory sensory system with the same sensing statistics as the data here recorded in Drosophila. So then, using this extended system, you can ask yourself, how well does it perform? So here, what I'm doing is I'm taking odors made of 50 components, made of 50 odorants. Then I imagine an olfactory system consisting of 50 receptors, 75 receptors, 100 receptors, et cetera, et cetera, et cetera. And then I go ahead and ask myself, how well can you reconstruct or decode the odor from those things? And what you discover is by the time you get to a couple of hundred receptors, the distortion that you have in the odor is very small. And conversely, if you have about a couple of hundred receptors here, 216 receptors in this case, then as you increase the number of components of the odor, right? So if you have an odor composed of 10 components to a couple of hundred components, you actually, the performance, the quality of your decoding, of your reconstruction of the odor barely changes. So that suggests that in the kind of regime of complexity of natural odors, given the kind of sparsity the natural odors have in odor space, a few hundred receptors is going to be sufficient for encoding all of the information. It's enough. So that's So the suggestion there is perhaps this explains the operating point for all animals. It's a very qualitative argument. If you have a few hundred receptors diffusely binding, sparse odor vectors, it's the case that, at least mathematically, all of the information about those odors can be stored effectively and efficiently in that body of receptors. So that's our first little piece of evidence. Okay. So that, again, this is speaking to, if you're going to distribute the information over many receptor types, how many receptor types should you have? I'm trying to argue that, you should have enough to store everything and no more than enough. So you don't need a few thousand, a few hundred is enough. Okay, that's the first argument.
Vijay, sorry to interrupt. Yeah. Just I missed it. Can you say again what the ordinate is, the y-axis, and what are the colors on the top plot?
The color is the fraction of responses that are above threshold. So what you can do is you can also, there are a couple of things you can change, right? One thing you can change is the number of receptors you have, and you can change their binding affinities. We're going to later make some comments about the binding affinities too. And the bigger the binding affinity, the bigger the binding affinity, the more responses you'll have. So what's being drawn here is the idea that so of course, the fewer the responses you have, the less information you have. We'll talk more about this later to decode what the odor is. So what's being done here is to decode the odor and then compute a distortion between the decoded odor and the original odor, and try to argue that as you get to a couple of hundred receptors, the distortion is low.
So the y-axis is some kind of error measure, but
yeah, it's an error measure. It's a normalized difference between the. Actually, now I'm suddenly confused as to why it goes up.
It goes to 2.
Yeah. And this thing is going over 2. So I'm now suddenly confused as to why it's going to 2. So there's some issue with the normalization of the y-axis scale. That must have been what was bothering you.
Okay, thanks.
Yeah, I have to check why it's 2. Probably that's a MATLAB typo of some kind. Okay, next. So I hope I've justified to you that a few hundred receptor types is enough. Okay, that's what I tried to justify. And we'll do this again in a more serious way in a minute. So, but given that I have a certain number of receptor types, I can now try, now that we've agreed to the operating point, that there's a few hundred receptor types, I can now try to answer the same question that I did with photoreceptors. I can say, well, you have a few hundred receptor types, and you can take your budget, which is, let's say, n receptors, the receptor neurons, and ask how many of them should be the red type, the yellow type, the blue type, and the brown type. What fraction should you commit to these different things? And so if I were to follow exactly the same logic that we did with photoreceptors, what would I do? I'd start with natural odor distributions. I'd put them through a sensing matrix. I'd maximize the mutual information with the world. And then I would ask, what proportions of these different receptors should I have? That's what we did before. So if I believe myself, I should do it again and see what I get. After all, we're still working in the sensory periphery. All right, so here's the framework. You maximize the mutual information, responses in the odor distribution. Sadly, unlike the visual system, people just have not got data about what natural odorant distributions look like. So we're going to assume the kind of sparsity that I talked about, right? And then assume that otherwise there's a Gaussian distribution of odorant concentrations around that sort of sparse structure, right? So that's just an assumption I'm going to make, right? Because for the life of me, we can't seem to find any data on what natural odorant distributions look like. What do animals experience? Okay, so we're going to assume that. Being theorists, we're allowed to. And we'll assume, again, to keep things simple here, a linear sensing matrix. So you have the concentration vector times the sensing matrix. That's what we've been talking about, that produces responses. And then you get to add noise. Now, remember, I said that all the receptors of a given type, let's suppose there are kj receptors of a given type, they're collected together in the second stage of the olfactory system, in these things called glomeruli. So then that's going to beat down the noise with a root n factor. So you have a root kj is the noise. So the more receptors of a given type you have, the more you can beat down the noise in the response. This is our setup, and we want to maximize the mutual information. Were I to write this out in detail, well, once again, here's the epithelium with the odorants. They go to the olfactory receptors. They get collected in the glomeruli. There's a response vector. More in math speak, you have the response is, the number of receptors times this response function, and then you get to, add some noise to it, and then you can work out and calculate by hand, because we made all these Gaussian assumptions for lack of actual experimental data. You can actually calculate by hand the input-output mutual information. So this is the same object that we discussed in the previous lecture, and I'm not going to discuss the details of exactly how we did this. There are some parameters that matter, including the abundances of each receptor type and the variances of the noise of each receptor. Okay, so these are the facts.
Another quick question. It's a detail, but I think it's important for the form of the solution. In principle, you could have two components of the noise, and one component would depend also on S.
Yes.
Right? It would be kind of input noise, but here you have just output noise.
You are completely right. So we have so this analysis could be improved by including the kind of noise that you said, and as well as including, by the way, dynamics and other kinds of things that I'm just completely ignoring. So in this section on the olfaction, I'm just trying to get to the broad operating point. I'm going to try to get roughly the right answers, as opposed to try to get the fine details, because the philosophy that I also advocate very much for theorists is you first need to find that your ballpark theory is ballpark correct. If it's roughly right, then you can work on details. But if it's not roughly right, then there's no point working on details. So I'm trying to be roughly right. There's almost no theory in olfaction. So I would like to build a theory that is roughly right, and then we'll work on details. So I'm going to try to argue that this is roughly right.
Okay, thank you very much. That's a very important point conceptually. Thank you.
Okay, so this is our response model. So what do we do? So go to town. So here's, as a theorist, I can do this. I can imagine two odor environments with different covariance structures. So let's suppose I have a panel of 1,000 odorants and 1,000 odorants. This is the covariance between the odorants. And an environment is defined, because I've got this Gaussian distributions of odorants, is determined by its covariance matrix, which odor molecules vary together. You can think about this as defining the odor objects, right? So if I have apple, well, when I have apple, certain kinds of odorants vary together and increase and decrease. If I have strawberry, other things increase and decrease. So the covariances between the odorant molecules in the world define the odor objects, is the way we're thinking. And here are two environments. These are two environments in which, there's large variances in some places along the diagonal, the large covariances in some places. In some cases, you remove one odorant completely, in others you don't, and things like this. And a generic prediction you can make from this kind of model is if you change at low signal to noise ratio in noisy conditions, which is typically the case of olfaction, the fraction of different olfactory receptor types, fraction of different olfactory sensory neuron types, which is in analogy to the different cone types that we discussed in the previous lecture, should be dramatically different in some cases between these two environments with different covariance structures. It depends upon the covariance structure. Depending upon what that is, you get this receptor that you should have a lot of, or that receptor you should have a lot of. Now, I would argue that olfaction is not like vision. Vision really has a very universal kind of statistics. But olfaction, the smells really change from environment to environment. So you might have expected that you would actually need to change then, to smell well, the fractions of different receptor types. If the theory is correct, you would want to change the fractions of different receptor types in different environments. So in fact, so when we first started this, we were like, well, that doesn't happen, does it? And then if you go look, it turns out that olfactory sensory neurons actually undergo regular apoptosis. So you replace the olfactory sensory neurons in your nose all the time, and this neurogenesis, they're replaced by new olfactory sensory neurons, each expressing a single receptor, right? That happens every month to us. So that being the case, you could ask, well, hey, then the theory predicts that the fractions of different receptor types should change in your nose month to month as your olfactory experience changes. So what do you know? There was a beautiful experiment done 5 years ago now, when these authors basically had mice of the same species that they exposed to different environments, right? They would put certain odorants in the water, and they found this pattern of change. So depending upon which environment the animals were in, receptors that were highly expressed didn't change much between environments. receptors that were expressed at low rates, in low proportions, changed a lot. They increased a lot or decreased a lot between environments. And also the variance between individuals was huge. So what we did is we said, well, can we reproduce this pattern of change? We can't reproduce exactly this figure, because to do this, we would need to know precisely a recording of the olfactory environment and its structure. And we would need to know the response properties of all 1,000 olfactory receptor types in the mouse to all of these odorants. And we don't have that information, right? And so what do? So the answer is, there is for 100 odorants and 60 neurons, this information is known. So we can make a model of this by saying, I consider 2 olfactory environments, each of which contains 60 odorants, 100 odorants, and they have different covariances that mimic the pattern experiment. And I have 60 neurons. Let me just see what pattern of change I see. And what do you see? You see the highly expressed ones, the ones that are predicted to be highly expressed, do not change between the environments. The ones that are predicted to be less expressed change a lot between the environments, and the variance is large between individuals. So in other words, I'd like to argue to you that the theory here reproduces the qualitative structure of the experimental results. And thus, if we had well-controlled olfactory environments and a complete map of how the receptors respond to these odorants, then maybe we can reproduce this precisely and explain these different proportions. So this is a controlled experimental test of the hypothesis that we were previously investigating using the fixed numbers of red, blue, and green cones. So this is a much more powerful thing because this thing is plastic. Okay, so I wanted to present that. Okay, so what is the time? So I had one more example to discuss here about how you could decode the combinatorial responses from this. But I think in view of the time, well, should I talk about this? Okay, maybe I will. I'll finish this quickly and then we'll take a little break. So then the next step is, okay, great. So we've agreed that you have ballpark, a few hundred receptor types probably, and that, the proportions of the different receptors change. What I didn't tell you is how such a combinatorial code could be decoded. And I'm going to try to argue to you that there's a very simple decoding algorithm that the brain could potentially use. And that also points to the statement that what you need is a few hundred receptor types that respond sparsely. So once again, this is our encoding model. You have receptors binding broadly to different kinds of odorants. They're collected in the glomeruli and then sent to the cortex. So here's a very simple, I'm going to explain to you a very, very simple decoding algorithm. And then I'm going to argue to you that it can be implemented by circuits of the form that you see in the brain. So what you could do is you could have, let's suppose that this is the odor here, right? It's a sparse odor. It's a high dimensional odor vector. These are all the concentrations. And this odor contains only 2 components, this one and this one. So here I'm taking things to be binary. You're either at high concentration or 0 concentration. Later on in the next slide, we'll consider something more continuous. Just bear with me for a moment. And like in the real nose, we imagine some random sparse sensing matrix. If you look at the actual sensing matrix, people respond that, yeah, there are correlations, but it's kind of, I give you a receptor and I can't a priori tell you which things it's going to respond to. It responds to this and responds to this. This one responds to this and responds to this. And there are some correlations, some odorants have more receptors responding to them. So suppose I have this random sensing matrix and multiply it times this odor, you'll find that these 2 receptors respond. So the observation I'd like to make is that a very, very efficient way of decoding such combinatorial representations is to use the power of silence. What I mean by that is actually receptors that do not respond in such systems are actually more informative about the identity of the original odor than receptors that do respond. Why is that? Well, you see, suppose I know that this receptor doesn't respond. Then I know that any molecule that binds to this receptor must be absent. So that must mean that this one and this one are absent. Right? So likewise, if this one doesn't respond, then I know this molecule and this molecule are absent. So just by knowing that the top 2 and the bottom 2 receptors don't respond, you can basically eliminate all of these odorants that are pointed to by arrows, leaving only just these 2 as being the candidate odorants in the odor. So just by knowing what doesn't respond, you can immediately eliminate almost all candidate odorants. So you can write down an approximate statistical theory, and that paper I referred to, there's a statistical theory that explains this. And it turns out that this is a very powerful method of decoding. You can extend this to include more continuous sensing matrices. So in other words, it's not just high or low or zeros and ones. It's a sort of more continuous thing. And you can use this competitive binding model as your response model. And you can show that basically you can use this elimination method to eliminate many, many odorants that are absent, and then to decode the actual concentrations that are present, right? So once you've done that, the ill-defined or undetermined decoding problem becomes a well-defined decoding problem, and then you can use the actual response functions to figure out the concentrations. So just to justify that this works, you can now do this, and using realistic numbers of receptors and realistic sparsity, what you find is a picture like this. So blue means it's very, K here on the y-axis is odor complexity. How many components are there in the odor? How many odor molecular types are in the odor? The x-axis is the number of receptors times the sparsity of the responses. You remember I told you that a given receptor only responds to some fraction of the odorant, so there's some sparsity. So you can do a plot using this kind of decoding algorithm, and you'll find that the yellow region here is where it decodes well. So basically the odors are not very complex, or you have enough of this S times NR, you'll find that you can decode very effectively. The peak occurs around here. So you can decode odors with about 40 to 50 components best when you have about S times NR, which is the sparsity times the number of active receptors around here. So it turns out S times NR is about 10 in humans and is about 9. The optimal S times NR is about 10. For humans is about 12, and for Drosophila it's about 9. So this is to try to justify to you the statement again, that if you agree that you talk about this kind of optimal coding, decoding, etc., you're getting an operating point for best performance that looks very like what's actually there in the system. Okay. So I submit that to you as another qualitative piece of evidence, or semi-quantitative piece of evidence that in fact this is the correct operating point and that we have a good idea of what's going on here. So one more thing that you might say, so what are the insights here? One is that a receptor that does not respond carries a lot more information than one that does. So there's a prediction from that. So an inactive receptor should eliminate all odors that bind to it. So you could test this, right? So what you could do is optogenetically or pharmacologically shut down some receptor types or their associated glomeruli, and the animal should behave as if some odorants are absent. There's another prediction here that there is a maximum complexity of decodable odors depending on the number of receptors and their sparsity. So you could just compare this across different species. By just comparatively doing this, you could test whether or not this kind of theory works. One thing I didn't say, because there wasn't time, is, I talked about this as an abstract algorithm. It turns out this algorithm using silence is implementable using a neural network that looks a lot like the actual structure of the olfactory pathway, the same things we've been drawing with a bunch of odor receptor types that are binding sparsely to many odorants. Those things are being collected in the glomeruli, the olfactory bulb, and then projected randomly to the cortex where you have some kind of associative network. So you can write down such a network that will implement the algorithm. So this was a kind of very rapid snapshot of an application of the ideas from the last lecture to the olfactory system. Now, I'm sure that was a little fast, so I would like to ask you for questions.
Could we go back to the slide? You have the scramble and measure. It's like many slides before. I think I missed something there.
This one?
Yeah, I don't understand this.
Okay, so measured is the measured response matrix, measured in Drosophila. Scrambled is it's the same measured thing, except I've scrambled all the entries. So this is a randomizer.
To mimic the sparse receptor structure?
Yeah, so basically destroying the correlations that are there. This is asking the question of whether the correlations that are clearly there within this sensing matrix affect things. So that's sort of scrambling it. So to keep the same distribution of responses, but you scramble the correlations. The Gaussian is a Gaussian random sensing matrix, what the theorist or the computer scientist would have written down. And the labeled line is a common interpretation that's made of the olfactory receptor sensing behavior. Just to say, oh, this receptor likes to respond to, eugenol and acetophenone and this and that, and doesn't really respond to anything else. So there's a common interpretation that's made by taking this kind of sensing matrix and thresholding it. So that you either have above threshold or below threshold responses. So these are four. So this is an interpretation, if you like, of this kind of sensing matrix that's often employed. This is a scrambling to remove the correlations, and this is the ideal Gaussian random sensor. And the intent here is to show that the labeled line interpretation really erases a lot of the information that's actually there in the true sensing matrix, that the correlations in the sensing matrix don't matter much for the decoding performance. That's what's being illustrated with these two lines. And that both the measured and the scrambled versions are actually quite close to the ideal Gaussian random sensor that packs as much information as possible into the small receptor repertoire.
Okay, thank you. So the take-home message here is scrambled and measured, they are same.
There are multiple take-home messages, but that's definitely one of them.
Okay. And could I ask another question?
Yeah.
So in cognitive science, neuroscience, we know like how olfactory connect to emotion memory in
yes.
Right. And so I'm wondering, do we consider there's back propagate prediction from the cortex? So that's how we store the memory.
Absolutely. So sorry, please finish.
Yeah, that's the whole question. Like, just like make predictions. I like strawberries. I'm more sensitive to strawberries. So
absolutely. So the reason I started working on olfaction was two things. One is it was so different from vision. I wanted to know if any of the ideas that I was thinking about actually even applied. And I think they do. I try to justify that. But the really cool thing is, olfaction is really closely tied to stuff like memory and emotion. So for example, near the Monell Chemical Sciences Center here in Philadelphia, there's an alleyway behind. And if I walk through that alleyway, there's some smell there, and I'm back in my school bus in Bombay. My feet don't touch the ground again, right? I'm on Muhammad Ali Road, in particular, in a district called Baikola. It's sort of this completely visceral feeling. And, famously, Proust described this kind of thing in his book, Remembrance of Things Past. And people talk about this kind of thing all the time. So how does that work? So that's my real interest. So the two things that I didn't talk about that we've done so far involve precisely thinking about how contextual feedback based upon your experience of the world proceeds to modify the representation of odors and separates them or brings them together and introduces their valences. So this is something that we've been actively thinking about. Part of the reason why I didn't discuss that today is that would be a full talk in itself, and I did want to talk about spatial cognition. And part of the reason that is a full talk in itself is then we have to start introducing notions of dynamics. So a thing that I completely ignored here is that the olfactory system is highly dynamical. So there's all kinds of dynamical time-dependent processes. There are oscillations, and the oscillations interact with the cortical feedback in various ways to modify the representation. So I've left out the dynamics. So there's a whole second body of work that we've done so far, which involves taking the second layer of the olfactory pathway, the olfactory bulb, using these Izhikevich neurons that we talked about, I guess, in the previous lecture. I think we briefly mentioned Izhikevich's method for modeling realistic dynamics of neurons, then building the entire network using that, and then studying the dynamics. So we've used that to study how olfactory feedback can lead to pushing. So suppose you've got two odors that are nominally different. They're made of different molecules, but they have the same valence in the world. They have the same behavioral outcomes. They mean the same thing to you. Then it turns out that you can record in the olfactory cortex, and it'll turn out that the representation of the odor in the olfactory cortex becomes more similar, right? The responses become more similar. And likewise, if there are two odors that are similar, slightly different strawberry, but one kind of strawberry, I don't know, smell means you're getting ice cream, the other kind means you're getting homework, then it turns out in your head it'll separate them, right? And they become different, more different. How does that happen? So there's this. So we have some theory explaining how you have random cortical. So let's suppose each behavioral context is associated with a random pattern of feedback, right? So there's some random pattern of feedback with the olfactory bulb. We have some study of how that random pattern of feedback can be used to separate or to bring together the cortical representation of the odor, make things that mean the same thing the same, as opposed to things that are the same thing in the world. What's important to us is not that they are the same, but that they mean the same. Very beautiful and interesting things with all kinds of philosophical implications. But I didn't talk about that.
Yeah. So Vijay, I have just a couple of very simple questions. One about the decoding and one a more general one. So about the decoding, first of all, if you go from this binary model to the graded model, then I guess you would have probabilities of kind of eliminating or not eliminating.
Yeah.
But and then you proposed this as a candidate decoder, but can you obtain it from some optimal decoder, from some maximum likelihood decoder, or?
That I don't know. So that's a weakness in this argument, that we don't have an argument that it's optimal, or rather what it's optimal with respect to, because you can always devise an objective function with respect to which your answer is optimal. So but we can't. I don't have an answer to that at all. So another subtlety to be mentioned here is Rava. As you well know, the projections from the second layer of the olfactory bulb to the cortex are thought to be statistically random. To literally decode the odor and to reproduce precisely the input odor vector here, these projections have to be related to this S hat, the projections to cortex have to be related to the sensing matrix. That's not the case.
That's right, have to be known.
Yes, even though there are papers that act as if by learning you can produce that. I won't mention those papers. So what our attitude, which I left out here, is that S hat can be random. And if it's random, what it produces here is some rotation, if you like, a random rotation of this odor vector, of the original odor vector. All you need is that every odor vector be mapped to a different vector in the cortex. You don't really care that it's the same vector. They have to be different and they have to be separable. So that's what we try to argue.
I see. Okay. And the other broader question was, here you're predicting or comparing to data actual number, like 12, the complexity is maximum when you use 12, what was it, S times NR or things like this. So since you're talking about actual numbers, then, how precisely the noise in the output of receptor and so on will matter. And how should I think about this? Like, how should I think about these
You're making a really excellent point. Every prediction here needs to have a theory error bar, because there's just lots of uncertainties. So for example, I say here that the sparsity is 0.04. Well, how do I know that? Well, there's a bunch of data, and that data, there's some thresholding that's been done. The experiments are noisy anyway, and maybe it's 0.04, maybe it's 0.05, or maybe it's 0.44044. And I've left out all of those subtleties. So that's a weakness in this analysis. So I submit this to you again as a ballpark thing, right? As opposed to if this had worked out to be 100,
right?
Then I would have said we're just off, and that it's not worth sweating the details until we sort of figure out what's really wrong here. I submit this to you to say it's roughly right, and then to really make a prediction, then you have to sort of make all those adjustments. You need to be a little bit more realistic about the response functions, get those in more detail. I told you competitive binding is a good description, but there are other phenomena actually like suppression. You can have odorants that suppress the responsive receptors without ever making them fire. They just suppress their responses to other receptors. So you got to include a few more things like that. And there's the dynamics that I mentioned that need to be included. So there's more work to be done. Olfaction is harder to deal with than vision. But what I'm hoping to submit is that you have to make a start. And using similar philosophies to what we were talking about earlier with vision, which is a much more developed science, you can make a start. And I believe that this is a start.
Okay, well, thank you very much. I don't know if there are other questions. Otherwise, if you want, we could take a break.
I should take a break. Can we make the break 5 minutes instead of 10?
Sure. So
because I have plenty to say for my lecture 2.
Yes, no, I mean, for me, it's fine. I think it should be fine for everybody, I hope.
Okay.
Okay, so see you all in 5 minutes.
In 5 minutes. So let me start up my this again. Whoops. Imagining space. Let me hide my controls. Floating meeting controls, and here we go. Great. So this is effectively my third example of the kind of broad ideas presented in the first lecture. And this concerns imagining space, about the circuits in your head that help you to think about navigation, planning, and so cognitive things like inside your head. So really, the question I want to go after here is, first of all, you got to think about, if you want to think about this, first, what is space? So since ancient times, we've understood sort of space as a sort of stage on which physical things are located. And people imagine space as, finite, infinite, flat, round, continuous, discrete, centered in the sun, and so on. We, at least as humans, are able to imagine inside our heads space as an abstract thing, right? Like the cosmos as a whole. Here's a picture of the cosmos, according to some Nepalese artist. And also we think about space as, relationships between entities. We construct spaces like this in our head, and we're able to manipulate them. We're also able inside our heads to move in space and between spaces. But even this has many variations, even in the human populations. So for example, Mesoamerica and Australia, there's always 4 cardinal directions, north, south, east, and west. But in India, sometimes people talk about 10 directions, north, south, east, and west, in between, up and down. So I'm just trying to emphasize that there's many ways in which your head can imagine space. So our modern scientific way of thinking about space, we're all scientists here, so we're heavily brainwashed to think in this way, is due to Descartes. And he gave us this modern notion of space as a stage for action, a coordinate system with a certain number of dimensions. And I've been using this kind of idea again and again and again in the talk so far. In the talk so far, a physical space in the world is 3 dimensions or 2 dimensions, or an abstract space of odor and so on. But it's always in terms of these coordinates. And of course, there are modern notions in physics like space can warp and mix and things like this because of relativity. But inside your head, space is an abstract thing. So animal life is completely defined by motion in space. If you'd stayed rooted in space, we'd be plants, and we aren't plants. So we think in space, and to plan actions, you have to learn the geometry of spaces, imagine how to get there, and imagine where you are at any given time. But again, as compared to, for example, these Cartesian maps and things like that, inside your head, here, any given location is basically an abstract pattern of neurons firing. Some pattern of neurons fires, and that's here. So in this sense, the patterns inside your head maintain what's called a cognitive map of your location. That's inside your head, and your mind is able to navigate this. So of course, it's a very, very interesting question to understand how this happens. So I'm going to think about this again in the same tradition that I used for both the vision section, the olfaction section. I'm not going to start with the data. I'm going to ask myself as a theorist, if you wanted to devise a neural system inside your head for imagining space, what would you do? And again, I'm going to demand that the system we imagine is somehow efficient, and and I'm going to use this efficiency as a guide for the organization of the system, and then ask how it compares to the actual data. So what is place? We already said that a given location inside your head is some abstract pattern of neurons firing. So you might say, so suppose I now want to make an abstract pattern of neurons firing to represent space, what could I do? Well, okay, so let's suppose the world was an 8-meter linear track, and you needed, because of the behavioral requirements, resolution of 1 meter within this track to support what you need to do in the world. Now, one way of doing that is to have something that resembles a place cell system, which is to say I divide the world into 8 1-meter blocks. Let's say neuron 3 is responsible for responding when the animal is in this location. So if my pointer is the mouse, you would hear brrrr, like that. Right? So this neuron 3 responds when the animal is in this region 3. So this is a system that requires 8 neurons to represent this world. And, these are a bit like place cells in the hippocampus, but, there's plenty of evidence now that place cells encode many things besides space. They encode, context, goals, they encode, smells and sounds, and all kinds of things are involved in defining which place cells respond where. Okay, so suppose I sought a more efficient way of representing space per se. What would I do? Well, here's an idea. I could divide the room at the biggest scale into two halves, the left half and right half, blue and orange, of 0 and 1, then recursively subdivide so that at the second scale, the blue neuron responds to the left half of the room, and the left half of the right half of the room, and the orange neuron responds to the right halves, and likewise you recursively subdivide 3 times. So now I have 2 neurons at the first scale, 2 at the second scale, and 2 at the third scale. Okay, so now I know, suppose the animal is at this dashed line, I would find that the 0 neuron, the 1 neuron and the 0 neuron fired. So this location I could talk about in this population code for space as being the location 0, 1, 0, and this location is 1, 0, 1. So this is clearly a binary representation of space. It's a base 2 number system. And notice that it only requires 6 neurons. It's a bit more efficient. Well, if the room was much bigger, or if the resolution we sought to achieve was much finer, you'd find that the gain of a binary system relative to the unary system that we were talking about earlier is much bigger. Right? I just took an example where the gain is small. Of course, you can use other bases like decimals. Now, animal navigation, by and large, is in 2 dimensions. We can also talk about things like bats in 3 dimensions, but we'll leave that for later. So suppose I ask about similar reasoning in 2-dimensional space. So here's a 2-dimensional room, 8 meters by 8 meters. Let's suppose, your behavioral requirements say that you need a resolution of 1 meter on each side. A place-like system would divide the room into 64 bins, and then you would need 64 neurons to report your location in each of these locations. Right? Great, so that's one possible system for encoding space. So alternatively, you could try to define the 2-dimensional analog of binary numbers. So what would you do? You could divide the room into 4 parts, colored here blue, orange, yellow, and green, and then recursively subdivide again and again, so that basically the blue neuron here responds on this lattice of locations. The blue neuron here at the smallest scale responds on a finer lattice of locations. So observe that now you can achieve a resolution of 1 meter on each side, and you only need 12 neurons to represent this. It's a massive savings compared to the 64. So if you thought that brain was expensive tissue and you should encode things with fewer neurons and make things more efficient, this is definitely better. And of course, you can use other bases like decimal, like base 10. Now let's take a little bit a look at some data. So in about 2005, the lab of Edvard Bainbrit-Moser showed that in the entorhinal cortex, there are so-called grid cells that respond in the following way. The square here is a room, and in the room, they've marked out hotspots of locations in the room where the animal was standing when the particular neuron they're recording from fired. So this neuron fired when the animal is physically located in places lying on a triangular lattice. If you go to the neighboring neuron, it turns out that that grid of responses will be shifted by a bit, so you've got different phases. And what's more, as you go deeper into the entorhinal cortex, the grids increase in size. And what's more, they increase in size in discrete modules. So there's a jump from here to here, and there's a jump from here to here. And each module, the grids have a similar size, similar period, and a similar orientation. So I ask you, doesn't that look a lot like this picture I was drawing earlier? This was the cartoon version, you might say, of this kind of picture. Of course, I'd have to draw, blue spots for the second neuron, or green spots for the third neuron, and so on. But there's a resemblance there. So that suggests that we should interpret the grid system in the brain as a 2-dimensional fuzzy neural number system for location. Okay, so now I'm going to try to build evidence for that by saying, well, my broad hypothesis all along has been that you should be efficient in your representation of space, right? So you should be efficient in the neural representations that you have inside your head. And here I'm going to take efficiency to mean you should, encode location with the resolution that you need while minimizing the number of neurons that you have to expend, right? Because that cost is going to increase the number of neurons. So I could ask, which number system, binary, decimal, etc., should the brain pick to represent space? Now, an important point to mention here is there is no requirement that the base of this number system be integer. Let me explain why. Normally, so in this neural context, the base of the number system is the ratio of the periods of these adjacent grids, right? And there's no reason why the ratio of these periods should be the same across scales. And there's no reason why it needs to be integer, because it can do whatever it wants. We need an integer base because we write down the numbers. But the brain doesn't write down the numbers, it has to represent the numbers. So it doesn't matter if it's a non-integer number system, if you like, or even if the number system is changing its base as it gets to larger and larger scales. It can do whatever it wants. It should do the most efficient thing, would be the hypothesis. So the idea is to predict the most efficient number system in this way to represent space, and then compare the result that you get with the data. Okay, so I want to immediately mention that there is a constraint that we haven't mentioned so far. So we're imagining a representation of space where, you have the red neuron firing here and the blue neuron here. At the second scale, the red neuron fires here and here, and the blue neuron fires here and here, that kind of thing. So if you have this system and the red neuron fires on all three scales, you know that the animal must be here. Now imagine an alternative system where the grids squish very quickly. So that, well, you have this grid, and then you have a compressed grid of a much smaller scale here. Then if the red neuron fires at both scales, you don't know if the animal is here or the animal is here. So because of this ambiguity, well, you can't decode it effectively. So you have a constraint that says that you have an ambiguity if the width of the firing region at one scale exceeds the period at the next scale. Now I'm being slightly loose here because real neurons don't fire in these nice square regions. Rather, they have these kind of more bell-shaped tuning curves and so on and so forth. So you can redo this analysis with more bell-shaped tuning curves and a distribution of these things, and you'll find a similar constraint that you get ambiguities if the width of the firing field at one scale exceeds the period at the next scale. So keep that in mind. Okay, great. So now I need to formalize the problem. So I'm going to formalize the problem in one dimension because it's easiest to write down, and then I'll tell you how to generalize to two or higher dimensions. So here is the dorsal end of the entorhinal cortex. Here is the ventral end of the entorhinal cortex. At the dorsal end, you have fine grids. At the ventral end, you have coarse grids. Let's call the period of the grid lambda I at each scale, and let's call Li the width of the firing field at each scale. Now, the parameters we're interested in are the ratios of the periods at each scale. So I can write an Ri to be the period at one scale divided by the period at the next scale. So that's the way I'm going to write it down. Okay. Now, one thing, a constraint we have is the resolution of the grid. So what's the resolution supposed to quantify? It's supposed to quantify how many pieces I can break the room into. That tells you how finely you can resolve the room. So that should be something like the largest period divided by the smallest period. And this is a product of these ratios of grid scales. So because that's a telescoping thing, lambda 1 over lambda 2 times lambda 2 over lambda 3, et cetera, gives me lambda 1 over lambda n. And that's the resolution. And this number, the resolution, should stay fixed. Now, how many cells do you need to represent space in this way? Well, you need many phases. So if I have a grid located like this, I need another one here, and I need another one here, and I need a whole bunch of different grid cells. And the number of grid cells I need is going to be proportional to the period divided by the width. Well, in two dimensions, it's going to be proportional to the period divided by the width squared, because it's going to be areas that count as opposed to linear dimensions. So in d dimensions, this is going to be raised to the power d, as is the resolution. This will be raised to the power d. Okay, so there's some coefficient here that involves how much redundancy you want in these in the responses. Fine. So we also have so the cost of the system, we assume, to be increasing as some function of the number of neurons. It may not be linear, it may be nonlinear, it may be whatever it is, right? But it doesn't matter what the function is. I know the cost is going to increase with n, with the number of grid cells. So our goal is to minimize the number of cells you use to make it as efficient as possible while keeping the resolution fixed. Well, we also have a constraint, and the constraint was that the width of the firing field at one scale should be less than or equal to the period at the next scale. Right? That was a constraint we set for unambiguous decoding. But according to this definition, the period at the scale I plus 1 is the period at the scale I divided by this ratio of grid scales. So that translates into constraint that the period at a given scale divided by the width of the firing field at a given scale must exceed the ratio of grid scales. That's the constraint we have. Now look what happens. To minimize the number of cells, I need to minimize everything on the right-hand side. Now these ratios, period over firing tuning curve width, is bigger than or equal to, for unambiguous decoding, the ratios Ri. So the smallest these numbers can be are these numbers Ri, the ratios of grid scales. So I can replace everything here by the Ris because that's the smallest they can be. Okay, so here's our problem then. We have Ri be the ratio of scales of adjacent grids. The product of these Ri, the product of these ratios is the resolution that we have to keep fixed. And the number of cells that I wish to minimize is a sum of these numbers. So I need to, so my problem mathematically is then to keep the product of a bunch of numbers fixed while minimizing their sum. In D dimensions, because we're talking about areas of different kinds, this would be, Ri to the D, and Ri, this would also be Ri to the D, because it involved, in 2 dimensions, it would be Ri squared, for example. What's going on? So you can solve this problem. It's easy to solve. And you'll find actually, that it makes a bunch of predictions. The first prediction is that all the Ri are equal. So in other words, like a base n number system, you'll find that the ratios of adjacent periods must be equal for every scale. The second prediction you make is that this constant ratio should be the dth root of E. So in 2 dimensions, which is for animal navigation, this is the square root of E, right? Where E is Euler's constant. So, this was work we did with a former student of mine, Xuexin Wei, who was actually, by the way, a undergraduate at Beijing University and is now a professor at UT Austin. And when we worked this out on the blackboard, that this answer should be square root of E, I told Xuexin, oh, that is so beautiful. That is so, so beautiful. But it's going to be wrong because there's nothing whatsoever in biology that ever works out this nicely, right? Just nothing. Okay, but wait and see. So first of all, before trying to compare with the data, you have to be a little bit more serious because I've used all these sort of fakey, perfect firing fields with no edges and, no tails and everything to be a bit more serious. So in a more Bayesian language, at any given scale, so let's suppose at scales bigger than a certain amount, you have a whole bunch of neurons firing, you'll find out that the animal is localized in like this. The black thing here is a probability distribution of where the animal is. This is using the full Bayesian technology for working out, given the firing of all the neurons, where is it likely to be? Then if you take the next scale, because it's periodic, you'll find that the response at that scale tells you that the animal may be here or here or here, and this is the periodic probability distribution telling you where it is. So if you combine these 2 scales and you multiply them, then, it shrinks. The total probability, once you multiply it out, is narrower, and that's how you shrink from scale to scale in a fully probabilistic language. Again, you have a constraint. If you shrink this too quickly, if the grid shrinks too quickly, you find out that you get side lobes, which then reduce the accuracy with which you can decode the location. So you have to, do that whole calculation, worry about that, and convolve the probability distributions and so on. Also, strictly speaking, if you are in 2 or higher dimensions, there's a lattice of locations where you could be firing. So you have to worry about the lattice and what the shape of the lattice and everything. Okay. So at the end of the day, you can work out what the answer is. And it turns out, here you go. So in the simple model we first described, the optimum is the square root of E, beautiful number. And here I've drawn 95% error bars, following the kind of thing you were saying earlier. There's some uncertainty as to what you want the theoretical prediction to be. So if you have the fully probabilistic Bayesian model, then the prediction is like this. So square root of E is like 1.6, and the fully probabilistic models, because of the tails in the distribution, winds up giving you an optimal ratio between scales, a base for this number system, if you like, of 1.4. Here are 2 data sets. This is from about 15 years ago. This data set is beautiful, but doesn't have that much data. This data set from the Moser lab has hundreds and hundreds and hundreds of cells for multiple modules. You can see there's a beautiful agreement with the theory. What's more, there's another data set by Julia Krupich et al. At Cambridge that came out a couple of years ago that has even more data, and that also agrees beautifully. Except it agrees, so it turns out English rats all have this scale ratio, Norwegian rats have this scale ratio. I'm not entirely sure why. No doubt this is why Britain left the EU, and protested the scale difference. But anyway, so that's but that's a minor detail because everything is within everything else's error bars. So this is an excellent match, and I would say evolution seems to have invented base n number systems long before people and optimized it for neural hardware. Really, what this wants to be is a base square root e number system for locations in space. So we seem to find that an efficiency principle seems to explain the organization of the complicated circuits supporting a deeply cognitive function. I think that's really cool. Okay, questions? Okay, so this was still very much in the spirit of the first talk and the early part of this talk, which I'm using an efficiency principle to try to understand the organization of a system. But what I really want to talk about next in the last part of my lecture series is to talk about, to go to the next step. You know, it's one thing to say it's all efficient and this, that, and the other. The next thing is to ask how it's formed. Because if you have such a circuit, either you have to wire it up during development or you have to self-organize it dynamically. Now, it turns out that the grid system has to organize dynamically. Why is this? Well, when the animal enters a new room, you'll find that it doesn't have a well-formed grid. Right? The cells don't respond on grids. And then as it explores the room for about 30 minutes, these grids begin to form, suggesting that the grid responses are actually self-organized through experience. What's more, it's clear that visual cues and things like that matter, because if you put a, let's say, a red marker in the room and rotate it, the entire grid rotates with it. So it's anchoring somehow to the boundaries of the world. However, you don't need that kind of sensory input. So for example, if you wander around, learn a grid, and then shut off all the lights and sensory deprivation and all of that, the animals seem to be able to do path integration, namely use their own sense of motion, their own sense of how fast they're moving, in which way they're moving, and how they're turning, and maintain their grids over long periods of time in the dark. So in other words, this suggests that path integration, internal path integration by itself provides an important cue for the formation and maintenance of these grids. So we would like to build, therefore, a model of the entorhinal cortex where it can dynamically self-organize a grid based upon its path integration, self-motion, and then integrate cues like visual cues and boundaries into it. To do that, you're basically treating neurons as a dynamical system that self-organizes. So how do you do that? So let me remind you of 3 approaches to doing this. The simplest way of doing this is what are called leaky integrate and fire models, where you have neurons that linearly integrate their inputs, but then leak charge. So the incoming currents cause membrane potential to increase. And then what you do is you declare by hand that if that potential reaches a critical threshold, you'll just settle it back to baseline. This is a very old model. It was invented by Lapicque in 1907, same year that Einstein invented special relativity. So it's been around a long time. And you know, it's a cartoon that suffices in many cases. You could go the exact opposite level of complexity. Hodgkin and Huxley won the Nobel Prize in 1963 for their beautiful work in which they figured out how to model all of the stochastic nonlinear biophysics of voltage-gated opening and closing of ion channels. And so you take the potassium channel, you take the calcium channel, you take the sodium channel, chloride channel, you put them all in, you put in the dynamics, you've got like you got a page full of equations governing all these things. You press go, you'll get realistic spikes. Every type of cell has different characteristics, all of this stuff. You'll get a very accurate description of the spike. And there have been people, communities in computational neuroscience who have felt that the way in which you describe the brain is to sort of build realistic models like this, build all the neurons you can, just put them all in and press go, and it will make you. And this was, for example, the general philosophy of the Blue Brain Project, that for a while, that you could just do this with cortical columns. Personally, I think that is unwise because the experience in physics suggests that if you start with quarks and try to derive the properties of iron, you're bound to fail because it's too complicated. That rather what you should do is at each level of analysis, you should get the correct coarse concepts and then use those to manipulate the system. So what I find in my lab to be a very effective intermediate between these two things is the approach of Eugene Izikevich, in which he basically gives what would be called in physics an effective theory of neurons. He has a sort of coarse-grained dynamical systems model of a neuron with about 3 or 4 parameters. And you pick the, if you, depending upon how you pick the parameters, you can get neurons that go blip, 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 brr, blip, 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 or brr, 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 brr. You can get neurons with all the kinds of different kinds of dynamics that we actually see in the brain. And so this is what we in practice use in my lab when we're trying to build neural network models of this dynamical systems kind, because it accurately will capture these kinds of things. But today I'm going to stick with the sort of generalizations of this leaky integrate and fire model because they are by far the easiest to handle, especially to make analytic progress with. So how do you organize, self-organize these grid systems? So there has been work for over a decade now that gives a nice model of how you would self-organize a single grid module. So let me explain that to you first. So these blue things are all supposed to be neurons in the entorhinal cortex, excitatory neurons in the entorhinal cortex. And you imagine there's a sheet of these things and that there is some external drive, possibly coming from the hippocampus, that's driving all of these neurons to fire. So in the leaky integrate and fire kind of description, S is the firing rate of the neuron, tau is the time scale at which the activity decays, this plus SR, well, if I bring it to the other side, it's a minus SR, describes the decay of the activity if the neuron is left to its own devices, and A of R is an external drive coming from elsewhere that's pushing these neurons to fire. So just imagine then, if you had this system, the neurons would all be going brrrr, that's in constant rate. Now imagine that each neuron inhibits its neighbors. If so, suppose this neuron here, this blue neuron, fires first. If it fires first, it's going to shut down a whole bunch of its neighbors. Well, but then a neuron that's a little bit away, outside its range of inhibition, will be not inhibited, and that will fire. And that will shut down a few more neurons. So in this way, if this was one dimension, you can see you'll get a lattice of neurons firing. And in 2 dimensions, you can show that if you set up this dynamical system, that the fixed point, the place where it stabilizes its activity, turns out to be a well-packed triangular lattice that you can see because, the first thing fires, it shuts a bunch down, then a couple more fire, and they sort of push each other around, and eventually 2 settle down. They'll be on a triangular lattice because that's the best packed, simplest way of doing things. Okay. So it turns out this kind of thing defines what's called a dynamical attractor, and the dynamical attractor as its fixed point has these kinds of triangular lattices of firing neurons. Now note that the lattice, the triangular lattice here, is not in the world. This does not describe locations of the animal in the world, but rather this is a triangular lattice of activity on the neural sheet in the cortex. So literally in the cortex, in this system, you'd find that you can put a triangular lattice and the neurons at the vertices of the lattice will fire. Now notice that something interesting has happened here. Before you had this lattice appear, this triangular lattice appear, you had a translational symmetry. Everything was firing. And now, by producing this triangular lattice, you've broken the translational symmetry. But now, how should you break that symmetry? You could place the lattice here, or you could place it here, or there, or there. You can move that lattice around, and clearly you can move that lattice around at no cost, as in the triangular lattice could have been placed anywhere. So what this does is in this system, there are essentially 0 energy deviations you can do. Namely, you can take this lattice and you can freely move it this way or that way. And the lattice is happy to move around. It doesn't cost you anything. So these are, this is well known in physics, this sort of situation where you take a continuous kind of symmetry and you break it to a discrete symmetry and you get these sort of free motions of the resulting pattern that you get. That can happen here. So the interesting idea of these authors, Burak and Feet, was to couple that free motion that you can have to path integration by the animal. So what you do is you take the external velocity signal. So you take the speed of the animal, which is encoded in some kinds of cells that respond to the same structure. And then you take the direction, the heading direction of the animal, which is also encoded in the responses of other cells in the animal. And then you couple those inputs into the model of the response. So you couple those into these various cells so that the pattern translates. So what you do is here, so that's being written down in equations here. There's the external velocity being coupled via head direction cells and speed cells in the activity of these neurons. So you couple that in to the dynamics. And then what happens is that this triangular lattice of activity is then coupled to the motion of the animal in the world. So if the animal moves to the right, the whole pattern in the cortex moves to the right. If the animal moves up, the whole pattern of activity in the cortex moves up. So you couple them together. So in this way, the neural sheet becomes kind of equivalent to a map of the world. You might say that the coupling produces an isomorphism between the neural sheet and the world, translating the pattern around. So let's see what happens. So suppose you're looking at this neuron here, the red neuron here. When the mouse is initially here, it's firing. That's the hotspot. Then the mouse moves to the right. And when it moves to the right, this it's no longer firing because the hotspot has moved to the right. But then the mouse moves some more and the hotspot will move back onto this thing and it'll fire again. So what will happen is this red neuron will fire when the mouse is here and when the mouse is here in the room. At two locations in the room, as this pattern in the head translates over the neuron in the cortex, this cell will fire. So in effect, this given neuron, that marked in red, will fire when the animal is located at a triangular lattice of locations in the world. So this is a trick for transferring the pattern of activity in the cortex into a pattern of activity for the individual neuron, depending upon the locations in the world. So this is the this attractor model for the appearance of grid cells. So what we want is we want to make multiple modules. Remember, I said that if you want to interpret the grid system as being a sort of base and number system, then what I really want is that I want to self-organize multiple grids such that the periods of the grids have some scaling relation between them. Remember, they have to scale by square root of E or something like this. Right? That's what we'd like them to do. So we'd like to know if there's a dynamical method of doing that. Now, one way of increasing the scale of the grid as you go from the dorsal to the ventral end would be to increase the scale of the inhibition that they experience. Remember, the way this worked is that you had this sort of inhibitory interactions, and the bigger the range of this inhibition, the bigger the period of the grid will be. The bigger you make this, if this thing fires, if you make a big range of inhibition, then the next thing that fires will be way over there. So you can make the grids bigger and bigger in their period by increasing the range of inhibition. So suppose you do that, you increase the range of inhibition in some continuous manner as you go from the dorsal to the ventral end. What happens? Well, it's hard to see in these pictures actually, but you'll find that the scale of the grid increases in a continuous way too. So if you increase the scale of the inhibition continuously, the grid scale will increase continuously. But what we want is a discrete hierarchy. We want the grid system to have one period in this module and then jump discretely, not continuously, to the next period and then to the next period in some scaling way. So how can you self-organize such a thing? Well, one way would be to very carefully during development fix it so that in this patch of the entorhinal cortex, this was the inhibition, you fix some inhibition length scale, and that somehow in the next patch of the entorhinal cortex, you get a jump in the inhibition length scale. So developmentally, you need to wire up the inhibition to be at bigger and bigger scales. But there really isn't evidence that that it works that way. And in any case, that would be very hard to organize developmentally. So it'd be nicer if there was a dynamical mechanism for sort of basically producing this kind of effect. Okay, so an analogy we might make is this was work done by a student of mine, Louis Kang, who's now a professor at Riken. And he's a condensed matter physicist. So he said to me, upon looking at this problem, this reminded him of something called the Frenkel-Kontarova model that appears in a standard textbook on condensed matter physics. So let me explain how this analogy goes. So you have a bunch of balls connected by springs, and the springs like to push things, to a certain separation apart. Then you stick this whole system within a bunch of potentials. So this is supposed to be the energy of keeping the ball here versus here, etc. And then what you can do is you can use this potential to move the particles apart. So the particles want to sit at the bottom of these energy landscape. So they want to be sitting at the bottom of them. On the other hand, the springs are trying to pull them together. So if you do this, then there's a push and a pull. And if you go through the system and kind of slowly increase the scale of which of these potentials, you'll find there are discrete jumps in how the system organized, in the lattice of where these balls are. So there's these discrete jumps that arise basically because there's a push and a pull, and the push and the pull compete and up to some distance the push wins, then the pull wins, then the push wins. So there's a change like that. So inspired by this, we can imagine the following model. So we have here the inhibition between grid cells pushing the grid to be further and further apart, to have bigger and bigger period. But then I need a pull to try to prevent it from separating. So you could do that in the following way. You could run between these different layers, between the dorsal and the ventral end of the entorhinal cortex, you could run longitudinal wires, right? Excitatory wires. So what you could do is, suppose I have this cell here. Imagine that it excites a bunch of cells at some neighboring region in the entorhinal cortex that's deeper in. So in fact, it turns out that anatomically such connections exist. And there's nice work by the Leutgebs at UC San Diego and by David Tank's lab in Princeton showing that these connections exist, but their purpose is not fully understood. So here we're proposing a purpose for them. So there are connections like this. And for our purposes, it doesn't matter very much whether one neuron here connects to one neuron in the next layer, or one connects to many, or many connect to many. It doesn't really matter very much. They all give a similar phenomenology. Anyway, what's going to happen is that if you run connections like this, then each one of these scales, each one of these sort of modules is going to try to force the next module to have exactly the same period. Why? If this fires, it'll try to encourage all of these to fire. And then if this fires, it'll encourage these to fire. So it'll try to enforce the same periodicity through these excitatory interactions. And if, on the other hand, the inhibition length scale is increasing from here to here, it's trying to force these apart to bigger and bigger periodicities. So you have a push and you have a pull. So there's some hope that what this will do is organize itself into a discrete hierarchy. And what do you know? It does. This, again, is a little bit hard to see, but you could find out that if you look more carefully at this, that there's a discrete hierarchy of jumps. So what basically happens is you have one scale, then it jumps to another scale and jumps to another scale. And of course, what those jumps look like, what that scaling ratio looks like, is going to depend on a balance between the excitation and the inhibition. So imagine that longitudinally along the entorhinal cortex, you have some excitation strength, and that horizontally, as you go along the entorhinal cortex, imagine that you have two modules here where you have L2 over L1 is the ratio of the inhibition range. Right, so you have a smaller range of inhibition in the first module, and then you have a larger range in the second module. So L2, the bigger L2 over L1 is, the more it's trying to the more it's trying to push apart the periods of the grids, and the stronger the excitation magnitude is, the more it's trying to coordinate the periods of the two grids at the two scales. So this is again a little bit hard to see. But if you look at this picture, what you're going to find is that this is overlaying the grids of the two scales, at scale 1 and scale 2. And around here, they have the same scale. Over here, they have the scale difference is 1.97. So depending upon how these parameters are organized, you get different ratios of periods. So in particular, we can simplify that a little bit and draw it like this. There is depending upon which phase you are in, namely what the ratio, the inhibition ratios versus the excitation strength, you can get different kinds of periodicities in these grids. So in other words, you can get different kinds of scale ratios. So as you go from scale to scale to scale, the jump in the periodicity, maybe 1.75, maybe 1.32, 1.28, 1.01. So depending upon how you arrange the parameters of the system, you can get different scaling ratios in this way. So that's cool. So this is a dynamical method of generating these kinds of discrete modules. And you can show that there's some experimental evidence for this. The basic prediction of this kind of model is that the relative orientation of modules would be preserved in individuals across different environments. That's because the coordination mechanism that's happening is that you have excitation from one module to the other, driving coordination between the modules. So that would mean that if you change the environment, the relative orientation of these modules needs to get fixed. And indeed, that appears to be the case in some data. And you can, and this is something we're continuing to study and discuss with you. Any questions? I'm getting a little tired. It's kind of speeding up. There must be a question.
Hi, Professor. So first of all, thank you. Have you tried to just to apply it also on 3D data. I know that there are some recent studies on bats and rats exploring a 3D space.
Wait, there's data on rats exploring a 3D space?
Yeah, I'm almost sure. Volumetric space 3D. Yeah, kind of 3D mesh. I don't remember. I can find it in.
All right. No, I would like to. I would like. I've been following the bat data off and on, but I haven't been following the. I didn't realize that there was now data on rats too. That's cool. I need to look at that. But so with the bat data, there's a bunch of things to be said. So at least the preliminary data on grid cells in bats seem to suggest that the grid system in bats was quasi 2 or 3 dimensional, because bats actually spend a lot of time crawling along the walls and they sort of fly off. So the initial data was not very clear whether I should be thinking about these things as 2-dimensional grids or 3-dimensional grids. So I haven't seen what the latest results on that are. That obviously complicates matters, whether you should think about this as quasi 2-dimensional or how you should be thinking about it. The same bat group from the Weizmann Institute, of course, has beautiful data now with very, very long tunnels, and in which kilometers long tunnels through which the bats fly. So the thing that I've been paying sort of most attention to in the last conversation I had with them was actually about the place cells, not the grid cells, because place cells people used to think of as having localized single place fields. But in their data, the place fields often have multiple place fields and often have different sizes and things like this. So there's clearly something interesting going on. With that larger data set, I haven't seen their grid data, but it's surely coming. If it's not already out and I'm not aware of it, then it's coming. And I'll be very interested to understand this. They don't have modules. As you can imagine, flying bats are very, very difficult. This is a tour de force, these experiments. So they don't have multiple modules yet. But to the best of my knowledge, I could be wrong. They're an excellent group and they keep producing new data all the time. So with a bit of luck, soon there'll be multiple modules and things like that also. But I haven't really engaged with that data yet.
Thank you.
But that's the right place to look. Really, really cool. Another interesting thing, by the way, is fish. Because fish also move in 3 dimensions.
Actually, I'm a birds guy, so.
You're a bird guy. Who do you work with?
Yeah, I'm good friend.
Okay, great.
Actually, Nako Molinovsky is one of the kind of collaborations in the same project. So.
Okay, great. So do you guys have program? All right. So do you guys have 3D data from the entorhinal cortex?
It's coming. I mean, it's coming. We have some data from barn owls, but it's going to be published soon, I hope.
Well, if you want to adopt me as your pet theorist, I'm very happy.
Yeah, let's do it.
No, that's
it's
really
not my call. I mean, I'm just a student, but yeah.
Oh, I don't know. You can tell you saw this really cool guy about this. We should adopt him as a pet theorist. But anyway, but seriously, I'm really looking forward to seeing that data on the birds because birds obviously live in 3 dimensions. So the place to test this kind of thing is you want to test in new circumstances. So I'm very interested to see that. There's some beautiful work by who's the person from the Tank Lab who moved to New York. I forgot his name for the moment, but he's been doing things with sort of birds that sort of hide seeds and things like that, and how their hippocampus stores all the different locations.
Dmitry Aronov.
Thank you. That's it. So I've been kind of off and on.
They recently published some 2D data, but 2-dimensional data.
Yeah.
Food catching birds. And,
he gave us a talk some time ago. So I think
also great book.
Yeah. So I think this sort of really wonderful information is going to come in about all of this in the next couple of years. So one of the reasons I'm presenting this is for any ambitious theorist, this is an area to really pay attention to, in my opinion. And you should get adopted by Ron here as pet theorist.
Sure. Thank you. Yeah.
Okay. I have 5 minutes.
You have as much as your vital energy will give you. But yes, we can go on for 10 minutes or something, sure.
Okay, so I'm basically at the concluding section of this talk, just to say one more detail. And mostly I will leave things unsaid. Ron has already highlighted a whole bunch of stuff that would be interesting to do that we won't talk about. but I just want to add one more thing. So what did we talk about? We talked about the grid system is somehow an efficient code for space. And I talked a little bit about how you could self-organize this kind of thing using excitatory, a competition between excitation and inhibition. That was going back to the, you need to be able to self-organize and readjust systems. And the last thing I want to talk about this will take 5-10 minutes, is I think that there's another way in which things self-organize and adjust and error correct and so on and so forth, involving, for the grid system, borders and landmarks. So let me explain that a little bit, because I started talking about sensory input and then said I'll leave that alone and talk about path integration. So if you believe this kind of attractor model of the formation of the grid system, then there's actually a problem at the level of the theory. So let me explain the problem. So we have these grids, they form beautifully, they move around. I tried to explain to you how you could sort of self-organize a discrete hierarchy and everything. But actually all such models have a problem in that they produce positional drift. That's because, the neurons are spiking, they're a bit noisy, the external inputs are a bit noisy, things are not aligned perfectly and stuff like this. And the net effect is that as the animal moves around, this model would predict that the grid will shift, will basically random walk its way. So there's a linear positional drift away from, with time, away from where it should be. So that would mean that your mental map of space just sort of drifts over time. So how could you solve this? Well, one way of solving this, obviously, is to anchor things to the world. Right? If the world is stable, you can anchor it. So, for example, the same structures in the brain have things called border cells. And border cells are known to fire when the animal is at specific locations along the boundaries. For example, here's a border cell that responds to the left edge of the room. Here's a room with a sort of barrier in it, and there's a border cell that responds at this location, and also this wall at the same kind of location, in the same orientation. So imagine that you want to use such borders, so those are like anchor points. You would like to use these borders as kind of anchoring your grid, your map of the world. Well, one way of doing it is to imagine some kind of plasticity, the issue that I said we'd come to at the very end. So you imagine a Hebb rule-like thing, for example, where neurons that fire together, wire together. What would that mean? You have a grid cell, let's say, that fires with the same phase as some border cell. So let's suppose you're at this location on this border, and this grid cell fires right there. It fires right there when you're there, and so does this border cell. Now, the border cells and grid cells are all mixed up together in the entorhinal cortex. It's entirely feasible for them to then, if they fire together, develop synapses together. So if that happens, what's going to happen is that, well, suppose this grid cell begins to drift. Well, this border cell, when it fires at this boundary, will go boom and shift the grid cell back into phase. That can be done. Let's check at the level of the model. It can be done. You set this thing up with Hebbian plasticity, and you'll find out that the black line is a positional drift in the grid, in the attractor model of the grid, without this kind of error correction. And you add this, and you do get error correction. So the boundaries can error correct the thing. So so this was beautiful work done by Ron DiTullio and also my former student Shui Xinwei. Now, how could you test this? Well, the real way to test this is to record from the border cells and the grid cells and see that their synapses, increase in strength in the advertised way, et cetera, et cetera, et cetera. But that's not been done. So here's another idea for a way of testing. So suppose you believe this is going to happen. Now
excuse me, before you talk about that, just if I could ask you, just to make sure I understand so here you assume that you have two kinds of cells. You have grid cells and border cells, and you couple them somehow to get...
Yeah, the border cells synapse onto the grid cells.
Okay, and then the border cells, you assume that so what's the model for the border cells? They just fire when the mouse hits the border of the room, like hits the wall?
So border cells, this is data in the top right. There are cells in the entorhinal cortex that fire when the mouse is, let's say, within 20 centimeters of the border of the room in a particular orientation.
No, I understand, but I'm saying in your model, that's how you model them. So you just say it fires when it's close to the wall, that's it. It doesn't have a dynamical...
So we did. So you can try to dynamically self-organize the border cells also, but in this model, they're not being dynamically self-organized.
Okay.
Actually, we have another model where they are dynamically self-organized, but let's not worry about it. Here, they are not dynamically self-organized. They just exist. So here's another test. So now what we can do is we can do a thing to test this. We can do a thing that never happens in the real world, only happens in things like Star Wars movies, that you're in a room and the room suddenly gets bigger or suddenly gets smaller. The walls squish. If you believe that there is this anchoring that happens, let's try to figure out what the prediction of such a model would be if you deform the room, because that's an easy experiment to do. You just do the recording you were always doing of grid cells, and then you change the size of the room. So what's the prediction? Well, you start out with this situation where you have a grid cell that fires with this phase, and you have a border cell, let's say the pink one, that fires at this wall. And you have another grid cell that fires with this phase, and this blue border cell fires at this wall. And let's suppose they're all connected to some degree. Hebbian plasticity would say that as time passes, the synapses between this red cell and this grid cell will grow stronger because they fire together. And the synapses between this blue cell and this grid cell will grow stronger because they fire together, namely at the right wall. And usually when you have this kind of plasticity, if you increase the strength of one synapse, there's a sort of homeostasis thing that happens where other synapses weaken. So then the cross synapse would weaken. Right? Certainly in the model, that's definitely what you would do. Right? So then these two are associated and these two are associated. Now suppose you stretch the room. If you stretch the room, what's going to happen? Well, here's this grid cell, which used to fire, let's say, at this location. But what's going to happen now that since you've stretched the room and the border cell fires, it's going to make the grid cell fire and it's going to shift it to the right. Likewise, this grid cell is going to get shifted in its firing phase to the left. So you predict here a boundary tethered grid shift. You predict that if this kind of phenomenon happens, where border cells interact in this way with grid cells, that if you change the shape of the room, you'll shift where the grid cell fires because of the interaction with the border cell. So does this happen? So famously, there have been a series of experiments by the London group. This is Castle-Barry et al. And the Stencil et al., that's the group of the Mosers. This is the group of Barry and Burgess, in which people have done these experiments. They took a room and they squished it or stretched it, and they recorded grid cells in such rooms. And what this is supposed to be showing is we went back and reanalyzed their data. This is old data. And we reanalyzed their data and found, indeed, that the locations of the grids were shifted depending upon the large boundary contact, or depending upon the last boundary contact. So, you get the shifted kind of reds move to the left and the green move to the right, depending upon which wall you last contacted. That's pretty cool. So that looks exactly like the phenomenon we were talking about. Now, why wasn't this phenomenon reported before? It wasn't reported before because these authors were reporting their time averaged rate maps. So what they did is they would average all of this data over time. So because they average the data over time and not conditioning it on the last boundary contact, if you average it, when you stretch the room, these grid maps look as though they've stretched. And if you shrink the room, they look as though they've shrunk. So the net effect is what they reported was that the grid cell map of the world looked like it was sort of painted on a rubber sheet. And depending upon whether you stretched or squished the rubber sheet, the map would stretch or squish. It's sort of covariant in that way with the stretching or shrinking of the room. But in fact, we would like to claim that that's an epiphenomenon coming out from averaging the data, that at least part of what they report is this boundary tethered grid shift that's coming from an error correction mechanism to prevent the drift of the grid. And actually, by the way, you can even show this happens in human behavior. You can do a virtual reality experiment where you give people virtual reality goggles, and then you tell them you have to walk, and there's a foggy room, they've got to pick up objects, then you stretch the room, you shrink the room, and you tell them put it back, and they wind up shifting their locations in this way. So that suggests, with humans you can't stick electrodes in people's head, but you can do behavioral studies, and it suggests that humans manifest this phenomenon too. So what are the next steps? I want to end with this. So there are many, many things to do here. So going back to Rava's question, well, ideally what you really want to do is, there's this whole circuit consisting of many different areas of the brain that talk to each other with many different cell types. And you want to self-organize the whole thing at the same time. That we talked about that circuit already in the last lecture. So you want to self-organize the border and the head direction responses and this overall network of different areas that are talking to each other. That's one thing that needs doing. Another thing that needs doing is all the experiments to date practically have been done in very simplified spaces, like little circles and little squares, and usually small spaces of about 1 meter squared or 2 meters squared, and occasionally very big spaces, like for bats and so on. So it's really important to somehow try to take all of this work to think about complex naturalistic environments. So, in vision, already for 2 decades, people have been talking about really trying to do vision in the context of natural images. And in what I discussed about olfaction, I talked about trying to push things about in olfaction, not to talk about responses, single odorants, but to sort of naturalistic odors, which are mixtures of things. So complex naturalistic spaces are important to look at. So for example, many of these experiments are done with rats and mice. And during the pandemic, a rat unfortunately took residence just outside our window because it was trying to eat all the bird seed in our bird feeder. And it made all these, it was clearly living underneath the snow and ice. So when the snow and ice melted, I went and investigated its burrow and took lots of photographs. And it's this very complicated thing, right? There are these passageways and little rooms and all kinds of stuff going around. So how does this all work in a more naturalistic environment? So actually, I'll tell you about that in a bit. So another thing is that it's now clear, for example, from work from the Tank Lab and elsewhere, that similar structures, the same structures in the brain, are used to imagine abstract spaces and high-dimensional spaces. So it's very interesting to ask the question, well, if you can use the same structure to talk about a 6-dimensional space or, some kind of abstract space of bird shapes or something like this, how do you do it? What's the map and how's it organized? Very, very interesting question. Another cool thing is about the difference between these sort of mental maps and your sensory experience. So your sensory experience is egocentric. It's relative to you. That's to my left, that's to my right, that's in front of me. But the maps inside your head are allocentric. The absolute, that's north, south, east, and west. So how does egocentric experience turn into allocentric maps, especially in complex naturalistic environments? So that's an ongoing project between my group and the group of Dori Derdikman, who's at a professor at the Technion in Haifa. Now, one of the reasons that's particularly interesting is, to me, the egocentric experience takes primacy. I really have a very poor allocentric sense of space. I can tell you things that are to the left or to the right. But there are languages, like, for example, these 3 Australian languages, Gugu, Yimthir, Kayadilt, and Tayor, and surely I'm pronouncing that badly, in which the speakers of these languages do not have a way of expressing egocentric direction. They always express things in an allocentric way. So you don't say the ice cream store is to the left, you say it's to the north, that kind of thing. So how does that happen? And so it's sort of very, very interesting. Anyway, this is an exciting and fruitful source of interesting problems for theoretical physicists. I should have said theoretical neuroscientists, but I said physicists because, inside my head, I think of myself as a physicist at the end. Okay, so that's that. So let me summarize. So overall, what have we done? We talked about distributed, efficient information representations of the brain, and I proposed to you that at least a piece of thinking about this could be understood by either maximizing represented information or minimizing the cost, depending upon what's most appropriate in bounded constrained systems. We looked at examples in vision, olfaction, and spatial cognition, but there's lots of work by lots of other people. I talked about my own work, but there's lots of other people to look at. And if you look at the reference lists in my papers, you'll find them. There's also work in audition, decision-making, and many other modalities. Definitely very interesting to think about. And I think it's a big problem. I think it's a very interesting problem to understand how and why information and computation are distributed inside your head in the way they are. Now, I'd like to highlight a weakness in everything I said. So everything I talked about was about information representation, right? About the average amount of information transmitted, the average amount of bits, this and that, and cost. But there's actually very little work on the word computation that we use. So if I really want to think about this as a computer, I'd want to think about this thing in terms of algorithms and dynamical change and self-organization, the procedures that are being used and how they're reorganized. And actually, there's almost no work on thinking about the brain in that way. So I think another sort of big open space to kind of borrow ideas from computer science and thinking about sort of the complexity of algorithms and the organization of algorithms to make them efficient and flexible, and to think about the whole brain and its architecture in that way. So I don't quite know how to do that, but every time I have a new student come into the group, I tell them this in the hope that they will tell me the right way to think about this eventually. So I'm going to tell you this in the hope that you will tell me how to think about this in that way eventually. So I'm going to end with this slide. whole bunch of open questions, which I leave open to you, the next generation. Why do we have such a diversity of functional units and computational procedures at each scale? What are the constraints on the costs of the functions of such engines, of such computational engines? Why are particular units and procedures that we see selected? So certain elementary computational procedures are employed as opposed to others. Why is that? And how and why? What are the rules that form the basis of the continuous reorganization of these kinds of circuits in response to behavioral demands? So these are big questions that I think we as theorists would really like the answers to. We'll stop there.
Well, thank you so much, Vijay, for this lecture. It's really wonderful, both on the detailed level and on this broader level. Thank you very much. I see that there are always already some questions. Claudius, do you want to go ahead?
I wanted to clap, sorry.
Sorry, I thought you were raising your hand, sorry. So if you don't mind, let's have a couple of questions. So I have a very narrow question. I feel a bit embarrassed even to ask such a narrow question after these big points, which were very interesting. Is that in all these models of these attractor models of spatial representation, the input is the velocity. And the velocity is also something that has to be computed by the animal.
Yes.
And I guess it could use a number of signals to compute, like vestibular proprioception, motor efferent copies, visual input. So do you have thoughts about that, about how that input is constructed, or what are the constraints or the noise on it?
No, absolutely. So several things. So there is definitely work on how those kinds of velocity signals are constructed, but I don't know it well. So I can't give you the sufficiently detailed answer. So there are cells in the hippocampal formation and the infrasternal cortex that encode speed and direction. There's head direction cells and there's heading direction cells. So that you can be walking this way, your head can be pointed this way. And these kinds of responses are documented. The precise nature of the computations, which are based upon the proprioceptive and visual input coming from the sensory areas that leads to the production of the signal, is not fully understood, at least in vertebrates. There is some interesting progress, detailed progress in fly, not surprisingly, where there's some interesting work by Vivek Jairam and others talking about how there are attractors for the I think head direction or the direction of the fly, and there's this torus that represents the direction. I think I saw one talk by Nahum Ulnowski talking about the appearance of such tori also in the bat. So there's some work known about that. So, but I think there's a lot more to do because there's actually multiple pathways. So if you look at all the ways in which all this information comes in, and in different, you can do this in different circumstances. You can do this with the eyes closed. You can do this based on smell. You can do this based on vision. You can do many things. So there's a thing going on here where these multiple streams are being integrated in some ways. And at least I feel that in what I've read, I don't see a complete understanding of that. And this is an interesting thing to work on. Possibly Ron has a comment because I see him nodding.
I'm just familiar with what you said. I mean, Dori, Nahum, and Yoram are kind of collaborating on the project that I'm working on. So, and I sent on the chat the 3D volumetric rats thing. I mean, it's just a preprint.
Yeah, no, I saw it.
Yeah, okay.
I never say just a preprint. Coming out of a
physics.
As far as I'm concerned, the preprint is the paper, and the rest is coloration. Anyway, it's a paper by Kate Jeffrey, excellent lab. So I will go look at that. Yeah, I saw your message. I saw it was a paper by Kate Jeffrey.
Yeah, I mean, if you work with Dory, so probably we'll work together sometime. Because I mean,
yeah, no, that would be good. I'm already Dory's pet theorist.
Some other questions. Shrangan, go ahead.
Yeah, thanks for the really interesting talk. I was wondering, because the hippocampus and all of that system is related to navigation in space, but it's also related to encoding memory. And I was wondering if from an efficient coding point of view, you had any thoughts on how like this region could kind of do both?
So I don't have an efficient coding kind of language for that, mostly because I've not thought about this very carefully. But it's very clear that your sense of space and your memory are closely tied. So, for example, if you think about episodic memory. There are different kinds of memory. First of all, let's point out. So episodic memory has to do with sequences of things. Declarative memory has to do with sort of naming things. There's different kinds of memory. So, but if you want to, for example, remember a sequence of objects, one of the most famous techniques for doing that is known in the Western world as the memory palace of Simonides, in which what you do is you take this long sequence of numbers or things you want to remember, and you imagine a palace, and you place the objects in the palace in different places. And then when you want to recall them in sequence, you remember the palace, and you walk through the palace in your head and look at the objects as you pass them and name them. And that's a very powerful way of remembering a sequence of things. So what that's clearly doing is it's harnessing our spatial circuitry and our ability to navigate to do something that wasn't really meant for, which is remembering long sequences of numbers or objects or something like that. So there's clearly a connection there. So this is something I would dearly like to understand, right? And going back also to another earlier question, I would also dearly like to understand, how olfaction triggers memories and modifies things too. So from my own sort of, if you like, from my sort of long-term plan as to what I would like to understand. So I've been working my way up from the sensory periphery. So I started in the retina, understood some things about it, learned a couple of things about other senses, some motor control, decision making. And I'm kind of working my way up to these kinds of integrated problems where there's space and there's memory, and there's space, time, and memory. And how do you put these things together? So, but I don't have anything more meaningful than that to say. Other than this is clearly a noted correspondence that people have between their spatial cognition and their ability to remember things.
So I don't know if there is any other question. If there's a burning question, please. Oh, I see. Yes, Yavuz, you have a question, please.
Hi, thanks for the talk. BioLake recently, or the recent few years, coming up with the idea of a coarse graining that relates to the neurons using their correlation, spiking correlation between them. Like they couple them, like in statistical mechanics, we just couple do the coarse graining according to adjacency. And he just redefines the adjacency according to the correlation, spiking correlation between neurons.
Yeah.
And try to come up with a coarse graining scheme with that. Under these circumstances, what kind of mapping or space structure should we expect out of this, or dynamical structure out of this, do you think?
So first of all, that's a very good idea to take, clearly, to take things that are correlated and lump them together so you get sort of a composite object that has things that are correlated. In terms of what kinds of outcomes that would give in terms of these structures and whether this would predict the kind of diversity of modules at bigger scales, I don't know the answer to that. I've not tried to do that myself. And I think Bill and his group haven't quite done things in that way either. So I can't give you a meaningful answer to your question. In terms of what we should expect, I'm not, this kind of thing is always full of surprises, right? There's the naive stuff, which is that, well, we know very well from physics, you take this dynamical system, there's some correlations, you take the correlated things, it could happen that, as you go up, the whole thing remains self-similar and keeps changing in a nice way, or it can happen that you go, there are sharp transitions, as in you come to some scale and all of a sudden the dynamics looks qualitatively different, you've gone beyond some critical scale in this renormalization group, as I'm sure it's called. Right. So what happens depends upon the details of the dynamics of the system. Right. Not all systems behave the same way. So which universality classes you see is one interesting question. And a second interesting question is what specifically happens, because we're actually interested in the actual brain. So we'd like to see the structures that come out there. So to neither of those questions do I know the answer, partly because I feel like possibly it's only now that the data is catching up to those kinds of techniques. It's only now the neuropixels probes give us the capacity to have 1,000 neurons. If we do that technique with 20 neurons, not much is going to happen. If you do that technique with 100 neurons, not much is going to happen. But with 1,000 neurons, 10,000 neurons, 100,000 neurons, maybe more would happen. So I think the blunt answer is that I don't know. I don't think if anybody else knows, but it's a very good thing to have techniques of this kind. That didn't actually answer your question. It just said it's a good question.
Thank you.
Okay, well, if there are no other questions, I should thank you very much, Vijay, on behalf of everyone. And these were really wonderful lectures. I'm sure everybody learned a lot. I did for sure. So thank you very, very much again for having taken part in the school and given these lectures and these discussions.
Well, thanks so much for inviting me. I had a very good time, and perhaps another time we'll be able to meet in person. So if anybody's in Philadelphia, let me know.
Definitely, with pleasure. Thank you again, and everybody see you tomorrow for the last day, I guess. Have a good night. Bye-bye.
Bye.